My name is Rosa Cossart. I am the director of INMED, the Institute of Mediterranean Neurobiology. INMED is a neuroscience center of INSERM and Aix-Marseille University that gathers 9 independent labs investigating the development and plasticity of synapses and circuits in health and disease. INMED is really a unique place where the link between early developmental programs and information coding and processing in the adult brain can be made. In this study, we investigated the development of GABA circuits in the barrel cortex. It's an important issue because GABA circuits not only provide inhibition in the adult cortex, they also shape in space and time cortical dynamics. In addition, an abnormal development of GABA circuits has been linked to most developmental brain disorders. We therefore examined the coordination of activity among GABAergic cells in the barrel cortex. We focused on the period of intracortical circuits development, which spans between the end of barrel formation to the start of active exploration. It's a period where interneurons undergo massive activity-dependent cell death. Understanding how cortical GABA circuits develop into functional networks is tricky for two main reasons. The first one is because, as in the adult cortex, GABA neurons are extremely diverse in terms of morphology and neurochemical content as described by neuroanatomists, but also regarding their physiology. Each subtype of GABA cell follows a different developmental journey and has a different embryonic origin. This diversity is present both in the adult but also in the developing cortex. The additional challenge when studying GABA neurons in the developing cortex stems from the fact that they have not yet acquired their remarkable characteristics. This is particularly the case for PV-expressing basket cells.
My name is Laura. I'm a postdoctoral fellow in the lab. I have performed all the in vivo imaging experiments to track the function and development of GABAergic circuits in the barrel cortex. To do that, we use calcium as an indirect way to measure network dynamics. And we use several transgenic mice lines that express a calcium-reported protein GCAM exclusively in GABAergic cells.
In particular, we use the GAD67 Cree mouse line that allows mapping interneuron activity without any prior focus on a particular subtype. As you can see in this movie, we observed that with this line we could not only image the soma of GABA cells, but also the ring of axonal activity provided by GABAergic cells around the somatic compartment of non-GABAergic containing cells. We further confirmed the identity of these perisomatic rings with immunohistochemical analysis. In this movie, you can see that GABA neurons are highly active and that they display recurring bursts of synchronous activity. This is a hallmark of most developing networks. You may also see that activity looks particularly patchy, with those patches sometimes coactive with each other. We observe the same patterning when the calcium reporter is only expressed in both prospective PB and somatostatin cells, but not when only somatostatin cells express the GCAM, which further indicates that prospective PB interneurons are the main components of GABAergic assemblies. These coactive patches merge into larger ones around the beginning of the second postnatal week, and now if we sensory deprive the pups by plucking the whiskers just after birth, we no longer see this evolution, which further indicates that sensory input is necessary for the broadening of GABAergic circuits.
I'm Yannick, and I'm a computational neuroscientist. For this part of the experiment, I used an iterative clustering approach to describe the latent neuronal population dynamics. In this graphic, we see the structure of the neuron's population dynamics. On the x-axis, we have the rearranged network events, and on the y-axis, the individual neurons and perisomatic rings. Here we color-coded each assembly that is determined by the contour's participation in it. What we observe at postnatal days 4 to 6 is that we have many small assemblies, as well as events that recurrently recruit the same assemblies. In contrast, at the beginning of the second postnatal week, we observe a decrease in the amount of clusters. However, the amount of cells and perisomatic rings that participate within a given cluster is increased. Additionally, we almost see an abolishment of multiple cluster recruitment in events. Color coding neurons and perisomatic rings contours on the physical imaging plane according to its participation in a cluster. We observe that they are also spatially segregated. Congruent with an increase in the assembly's functional domains, we also observe a spatial broadening.
Therefore, perisomatic GABAergic circuits, despite the protracted emergence of the parvalbumin marker, display early structured dynamics. This is in contrast with somatostatin interneurons that show broadly correlated firing. These assemblies are regulated by whisker inputs and altered by sensory deprivation. They form a transient functional scaffold, tailoring intracortical dynamics to the external world prior to active exploration. In particular, we know that they regulate locally interneuron survival, as elegantly shown in the companion paper by our collaborators, Duane and colleagues from the lab of Natalia Di Marco at Cornell. We believe that a disruption of this functional template by genetic or environmental factors may well serve as a substrate for sensory perception disorders.